Thank you. I'm delighted to be here, and I'm delighted to see such a great audience, whether you had to be here or not. Yes, as I was telling everyone, I've been at Ohio State for 22 years, and this is my first time at Ohio Dominican. So it's a great campus, and I'm looking forward to meeting some of you after the lecture. So before I start, I read some passages about Albert the Great, and I was really fascinated by how he, at the time when he lived, that he basically knew everything, like was said before, specifically every science that was known to man at that time. And not only did he know everything, but he contributed to all of these sciences. And these included areas like anatomy, anthropology, astronomy, biology, botany, chemistry, dentistry, geography, geology, medicine, etc., all the way down to zoology. All right? In addition, he was a leader of the Dominican order. He founded Dominican convent schools in Germany and then went on to study and lead the order in Italy, other cities in Germany, France, and finally settled in Cologne. And you know, one thing that struck me was that he actually not only taught, but also embraced both science and theology. And that is something that we need more of in the world today, I believe, especially if we think about ongoing issues in the world like climate change, things that we need people to pay attention to right away. So with that, let me just tell you about our work at Ohio State. So as my title says, we use light to start the reactivity of molecules. So the idea is that you have a molecule that is what we call the ground state, which is in its normal state, but it can absorb the energy of a photon of light, and that places it in an excited state. So now the molecule has gained energy, and it can use that energy to do reactions that we couldn't do from the ground state. All right? So this is important because now we can use the energy of light to do whatever chemistry we wanted to do, whatever reactivity we wanted to do. So this is how we use solar energy. So for example, we have molecules, and we do this in my group as well. I'm not going to tell you about this today, but we have molecules that transform the energy of the sun into electricity, or the energy of the sun into the form of chemical bonds or fuels. So this is important because we can now harness sunlight and use it as fuels. But today what I'm going to tell you about is applications that have to do with therapeutics and diagnostics. Right? So if we think about therapeutics, one thing that comes to mind is a disease that is still very much a problem in our society, and that is cancer. And these are some recent cancer statistics. And so it turns out that in 2012, 14 million people were diagnosed with some kind of cancer. And it's predicted that in 2030, there will be 21 million new cases. And if we look at the different types of cancers, the ones that are highlighted in red here are still quite problematic in that the 5-year survival rates are quite dismal. For example, lung, esophagus, 17%, stomach, 28%. And so there's still a long way to go in terms of having better therapeutics, early diagnostics, and intervention. Okay. So if we want to target cancer, the first thing that I question is, what is the difference between cancer cells and normal cells? And so here are some key differences. And so in cancer cells, we have increased rate of cell division. They're unresponsive to something that we call apoptosis, which is cell death. So when the cell's not doing well, it triggers apoptosis or it kills itself. Cancer doesn't do that. It erratically mutates, reproduces mutated cells, and normal cells don't do that. And another important aspect is that cancer cells can easily detach from where they are, and they can travel to other parts of the body. And that's what we know as metastasis, right? So that's a terrible complication of localized cancer, all right? So if we think about, these are the properties that we see in cancer cells versus normal cells, but how can we use these different properties to target cancer, to kill the cancer cells somehow? And so when we do that, we need to think about what are the properties of the cell that give rise to these different properties. And one thing that happens in cancer cells is that all cells have enzymes that do cellular functions. And in cancer cells, some of these enzymes are what we call upregulated, or there's excess of them being produced, and that's what causes some of these problems. So these enzymes, for example, there's a class of enzymes called tyrosine kinases, and basically they sustain proliferation of the cells. So those are overexpressed in cancer. There are also these P450 type enzymes, and these allow these cancer cells to suppress evading growth, so the cancer cells keep growing. In cysteine proteases, they actually, when they're overexpressed in cancer, they allow the cancer cell to escape and migrate to other areas of the body, all right? So if we think about this, in cancer, these enzymes are overexpressed, so we may be able to target specifically those enzymes in cancer to kill or mitigate cancer progression. And so that's what what I'm going to be talking about, and I'm going to be coming back to this diagram. I just wanted to show you how we're planning to do this. Okay, so when we think about cancer drugs, we often think of some big organic molecule. And it turns out that I just want to point out that there's a whole other periodic table. So for those of you who are not chemists, organic molecules are composed of carbon, nitrogen, oxygen, hydrogen, pretty much for the most part. And we need to keep in mind the fact that we have a lot of other elements in the periodic table that we can use to address cancer. And in particular, the ones shaded in blue here are metals, transition metals, that are actually essential elements in our bodies. All right, so a lot of times we are afraid of metals, but that's not always the case. I mean, yes, there are metals that are bad for you, but it depends on what's around the metal. And this is something that we can control, okay? And as a matter of fact, if we think of cancer drugs that contain metals, there are 3 main drugs that contain metals, and these are platinum drugs. And so cisplatin was discovered in 1978. Was discovered in the late 70s, and it was approved by the FDA in 1978. And since that time, there have been only 2 other metal-containing drugs that are very similar to cisplatin that have been approved for use for cancer treatment. So these drugs, it's astonishing to me that since 1978, only 2 other metal-containing drugs have been approved. These are the only metal-containing drugs approved for cancer treatment. And they're currently still being used. About 50% of cancer patients get one of these 3 drugs. But we know that they're terrible because they have terrible side effects. And that's because they're not localized to the tumor. They actually go throughout the body, and they act on healthy cells as well as on cancer cells. And so this is a big problem, but they actually work pretty well themselves, otherwise they wouldn't be used. So that's the best we have right now. And so it's important for us to continue to look for newer drugs, metal-containing drugs, that mimic the action of these types of molecules, but that don't have the side effects, okay? And so I'm going to be talking about a different metal. And so those 3 drugs they have platinum in them. Where's platinum? Right here, this is platinum. I'm going to be talking about ruthenium-containing drugs. So ruthenium is right here. And just to show you that ruthenium has been very popular, and I know you can't see all of these molecules, but this is different molecules that contain ruthenium that are currently being used in clinical or preclinical trials for cancer treatment. And one thing that I'm going to be talking about today is using light to activate these drugs. Because again, all of these, if you give them to the patient, they're going to be traveling throughout the body. And again, they're going to be toxic also to normal cells. And it turns out that there's one drug currently entering phase 2 clinical trials, that it has a ruthenium center right here, and it has what we call ligands around it. And it has been very successful because on mice models, it's not active in the dark at all, but when you shine light, that's where we activate the drug. And so that means that we can shine light just on the tumor site, and that's the only place where the drug will kill cells, all right? All right, so the use of light for cancer treatment is not a new concept. It's actually currently being used in the clinic, and it's useful for the treatment and sometimes superior for many cancer, especially those that are inoperable for various reasons. Maybe the cancer is near a blood vessel, or the cancer, actually head and neck cancers is very important because operations sometimes are very difficult because you could damage some other tissue like the vocal cords, etc. And so one problem with these systems is that they all work via the same mechanism. So the drugs themselves are not active. The way they work, and it turns out, so let me back up a little bit, photofrin right here is made up of this type of molecule, hematoporphyrin, and that is the only drug approved for what is called photodynamic therapy, which is what I just explained. You shine light to turn on the action of the drug. And so all of these drugs work by, you excite the molecule with light, you populate what we call a singlet excited state, which then decays to what a triplet excited state, which is very long-lived, that lives a long time. So oxygen has time to diffuse and transfer energy to generate singlet oxygen. And singlet oxygen is actually the species that kills the cells, all right? So these have been working pretty well, and those are the ones that are currently being used in the clinic. But one problem is that these molecules, they don't enter the cancer cell, they stay outside. They make singlet oxygen outside the cell, and that means that they cause necrosis, and necrosis causes inflammation, which is very painful to the patient. And so what we would like to do is to have compounds that also make singlet oxygen, but that go inside the cell to cause apoptosis so that it doesn't cause inflammation and it's not painful to the patient. Okay, so if we back up a little bit and summarize all the stuff I just told you, basically what we have are compounds that we want to excite with light and put them in different excited states. In one case, we're going to release drugs after we shine light. In other cases, we're going to produce singlet oxygen with the molecules entering the cells. And an important part of what I want to tell you at the end is that we can combine these two features into a single molecule. So we're going to have one molecule that both delivers a drug, but also generates singlet oxygen so that we can address the cancer by two different mechanisms, kind of like having a drug cocktail, but they're inside the same molecule. Okay, so just quickly, first of all, I'm going to start telling you about compounds that deliver drugs with light, and then I'm going to tell you a little bit about compounds that generate singlet oxygen, and then the combination compounds and also compounds that we're going to selectively send to the tumor to get further selectivity of cancer. Okay, so in the first part, how are we going to deliver these drugs with light? Well, I talked about ruthenium compounds. So these are two ruthenium compounds that we use. And this is what we call the cage. And this is kind of an old terminology. It doesn't seem applicable, but basically the way the reason this is called a cage is because the drugs are bound to the metal, and that means that they're not active. And when we shine light, we release them or basically uncage them. That's where that term comes from. okay? And so these are 2 cages, and they're not toxic, whether before or after. And when we're doing this in water, when the drugs comes out, we make this bis aqua compound for the chemists in the audience, all right? So going back to this diagram, we want to inhibit these, I'm gonna concentrate on this side for my talk today. So we want to inhibit these enzymes that are overexpressed in cancer. So let's take a look at what the inhibitors look like. And so these are structures, the chemical structures of some of these inhibitors. And where I have the red arrows here, this is where these molecules can bind to the ruthenium metal. And so there's different kinds. Oops, sorry about that. There are different kinds. There's one kind where we have a nitrile group that can bind to the metal. And then in the other cases, we have this pyridine groups that can bind to the metal, right? And we started looking at these inhibitors first, and the reason for that is because we know from model compounds that these get released with much higher yield than the pyridine-containing ones, okay? And that is shown here for these model compounds. So for when you have nitriles, you shine light, you have 20% conversion and 20% release of these nitriles, versus when you have pyridines, you only have 0.6% release. All right, so we started with the nitriles first, and these nitriles, they inhibit the class of enzymes called cysteine proteases. So cysteine proteases, what they normally do in normal cells is they're involved in cleaving proteins, all right? So this is how they work. In cancer, they're overexpressed in cancer, so they come to the surface of the cancer cell and they're facing the outside, and what they do is they start cleaving proteins, but they're collagen outside the cancer cells. And what that does is it makes the cancer more porous, if you will, and it allows the cancer cells to escape. Okay, and so we would like to inhibit this because that would inhibit metastasis. And so the way that we're doing this is we have the peptide that binds to the enzyme active site, and then the nitrile part actually reacts with the enzyme thiol in the active site and inhibits it by having this covalent bonding to the active site. Okay, so we started this work in 2011, and so we basically, you take your starting material, you combine it with the inhibitor to make the cage ruthenium compound. So we have 2 inhibitors bound here, and what we can do is then shine light and release the 2 inhibitors, okay? And so we can follow this by monitoring the changes in the absorption spectrum of the compound. So if we look at this complex, when we look at how much light it absorbs here at around 400 nanometers. As we start shining light, you see that this decreases and we get an increase in this peak after 1 minute, and then if we keep shining light, we get this peak. So this spectrum corresponds to the product, and what we do is first the molecule absorbs 1 photon, it releases 1 inhibitor, and then it releases the second inhibitor, okay? And we can actually see what happens in the presence of enzymes. And so if we do this in the presence of enzymes, this is the enzyme activity as a function of concentration. And so this is the inhibitor by itself, not in the ruthenium compound. So you can see that it requires about just under a micromolar concentration to inhibit 50% of the enzyme activity. When it's bound to the ruthenium compound, we actually decrease the activity by an order of magnitude. Now it requires 10 micromolar to get the same activity. But when we shine light, we actually recover all of the activity because we're releasing the inhibitor, okay? So this is just in vitro, just the enzyme in clean enzyme. We can do this with cells. We have done this with cells. And so now we have a different inhibitor. This is this inhibitor for one of the cysteine proteases. This inhibition now, it only takes 35 nanomolar. So it's a very small amount of the inhibitor is required to inhibit the enzyme. And again, we made the ruthenium compound. And what this is shown here in the cell work, where you see here the green light, the green spots, that means that there's a lot of enzyme activity. So this is ruthenium compound in the dark. If we shine light for 15 minutes, we can see that we inhibit all of the activity of the enzyme because we're releasing the inhibitor. And this is a control compound with just acetonitriles. Even if we release the nitriles, nothing happens because they are, it's not an inhibitor, okay? So this is a normal two-dimensional cell culture that most people use, but because we were trying to look at this collagen, breaking up of the collagen, we actually then turned to a different collaborator, Bonnie Sloan at Wayne State, who actually does three-dimensional tumor models. And that is a much more realistic model when we're trying to see this collagen degradation. And so Dr. Sloan actually works with triple-negative breast cancer, and that's a very lethal type of breast cancer that's very difficult to treat. And this is the inhibitor for that. And so you can see here's the nitrile that we're coordinating, binding to the metal. And again, this is the 3D tumor. So the control, you see a lot of enzyme activity, the green again. In the dark, you see a little bit of decrease of the enzyme activity, but when we shine light just for 30 minutes, you see a lot of decrease. And so we're very excited by these data because we have shown now that these compounds are not toxic in the dark, they don't do anything, and they only release the enzyme when we shine light. Okay. One thing that I didn't tell you about is something that we call the wavelength of light, or the color of light required to do this chemistry. And that's actually a very important feature. All of us have put a flashlight on our hands, right? And the only light that you see come through is red, right? So only red light really passes through tissue efficiently, effectively. And so we would like compounds that absorb red light. All of the ones that I told you about actually absorb blue, green light, but we need to shift where they absorb light. They need to absorb in the red so that they can be more useful. And it was really not believed that ruthenium compounds would be able to do this, but I just want to show you that recently we managed to do this. And so this is my postdoc Malik, and he discovered this particular compound. So it has one nitrile here that we can use for drug delivery, and he changed this ligand now, and he used this, what we call an ACAC ligand, and the absorption spectrum of this compound, you can see how different it is from that of the other ones. And so red light is around here, around 650 to 700 nanometers. This is actually near infrared, and both of these are very good for light penetration. And so we're very excited now to get this compound to try it with our cell work and our inhibitors. Okay, so let me shift gears a little bit. I showed you this wheel before, and so we only concentrated in this area, but as you can see, most of the other inhibitors have pyridines. So one big challenge was, how do we get our compounds to release pyridine efficiently, or some of these pyridine-containing drugs efficiently? And so a postdoc and my graduate students, Jessica and Brian, a couple of years ago came up with this strategy. And so now we have a different scaffold or architecture around the ruthenium. We have what we call a tridentate ligand and a bidentate ligand. And what we did is we kept this constant, but we varied the bidentate ligand. And we were monitoring to see if they release pyridine when we were shining light. And it turns out that if we have bipyridine here as the bidentate ligand, we didn't see any ligand dissociation. But when we had this dimethyl BP, we actually saw 16% ligand dissociation. And that's very nice, and you can see that here. These are the changes in the absorption. We're actually shining red light here. It works well with red light. And within just 15 or 30 minutes, you can see full conversion, full release of the ligand. So one question is, why is this happening? Why does the dimethyl BP work and not the BP? They're very similar complexes. And we think we understand this because these methyl groups are pointed towards the metal, and that actually distorts the geometry of the molecule. And so when you do that, you can see that here, this is the models. If we look down this nitrogen-ruthenium bond, and you can see this tridentate ligand is flat. We're looking this way, so this one is vertical. This is nice and octahedral around the ruthenium, but when we go to the dimethyl BP, you can see all of the distortions around the metal. And so basically that weakens the bonds around the metal. They're still very stable in the dark, but it allows us, when we shine light now, to be able to deliver the pyridine ligand. And basically what I said, we can say in different ways that chemists like to talk about. And there's a state that is anti-bonding, just for the chemists in the audience, that is ruthenium ligand anti-bonding. And basically these distortions lower the energy of this state that leads to dissociation below the metal to ligand charge transfer. So we understand how this is working, and now we want to harness more of this type of properties to be able to do these better, okay? So now that we know how to deliver pyridines, the question is, can we deliver a drug? And the first drug that we tried is this drug right here, ABI. So ABI is currently used for the treatment of prostate cancer. And as you can see, it has a ton of side effects: joint swelling, pain, diarrhea, cough, etc. And so again, these are due to the fact that the drug acts everywhere in the body and not just at the cancer site. And so the way that this works, that this inhibitor inhibits this P450 enzyme. It binds to iron in the enzyme active site. So we wondered, since this nitrogen requires to be free for inhibition, we thought that if we bound this to our ruthenium, then it wouldn't be able to inactivate the enzyme in the dark, and only when we shine light become activated. So we did this reaction. We actually made this compound, so with the tridentate ligand, the dimethyl BP, and we added our drug now. We made this compound, and we showed that when we shine light, we release the drug. This is shown here, changes in the absorption spectrum as a function of irradiation time. And we see that this happens with, it's not a very good yield. It's about 5% yield, but it's working, it's happening. And so we went on to do some cell work with prostate cancer. And this is shown here. So in the blue is actually the drug by itself. So you can see as we increase the concentration of the drug, we get more and more cell death. When it's bound to the compound, to the ruthenium, which is the black, you actually don't see much cell death at all, even if we increase concentration. But when we shine light, which is the red, we basically get the activity back of the free drug. Okay, so we're very happy with this new scaffold for the delivery of these nitrogen heterocycle drugs. I think that it can be used to deliver a lot of drugs, not just for cancer, but for other applications. And as you can see, this came out, was published last year. And these are just two different studies that were published by different people in the world using our ruthenium scaffold now to deliver drugs. And this is actually very rewarding. I think most of us scientists, we publish our work basically so that other people can use it. So it was very rewarding to see that other people are indeed, first of all, reading the literature, right? And second, actually using our scaffold, okay? All right, so quickly, I'm gonna move on, and I may skip some slides, but I'm gonna move on to compounds that make singlet oxygen efficiently, and then putting them both together, okay? And so now we get away from ruthenium a little bit, and these are what we call dirhodium compounds. So these are compounds with two rhodium metals, and these have for a long time been known to have anti-cancer properties. You can see some literature here from the 1970s. But again, they cause toxicity everywhere else. And these compounds were being studied by Professor Kim Dunbar at Texas A&M, and she's a good friend of mine, and she knew that we wanted to use light to activate anti-cancer compounds. So we talked and we decided to try to collaborate on this issue to try to activate these types of compounds with light. So what we did is we made a whole series of compounds. This work has been going on for a long time. And we start from our dirodium compound, and we can put different ligands on it. But an important ligand is this one right here, that's featured here. It's called a DPPN ligand. And this DPPN ligand is actually the ligand that's important for making singlet oxygen, which is what kills cells. And so we made a whole series of these compounds, and it turns out that they're not very toxic at all in the dark. So we measure that it requires from 50 to 200 micromolar to kill cells, 50% of the cells in the dark. But it turns out that when we shine light, we get about 25-fold increase in toxicity. And that is because we are generating singlet oxygen in pretty high yield. And one thing that we wanted to know with these compounds was where they're going in cancer cells. Where are they going inside the cells? And one problem is that these compounds are not emissive. They don't emit light, so we can't just follow them directly. And so one thing that we did was to bind an emissive drug to one of our dirodium compounds to be able to follow where they're going in the cells with confocal microscopy. And so we did that. So we attached this BODIPI dye that is highly emissive. And what we do here is we use stains that are known to go to certain areas in the cell. And so in green here is the emission of our compound. And here is a stain that we know goes to the nucleus. And so then what you do is you overlay the images and you see if they are co-localized. And you can see here that the green dots are well outside where the nuclear stain is. That means they're not going to the nucleus. What we do see is that they go to the lysosomes in 40%, and 31% of them go to the mitochondria. So this is work that we did a couple of years ago, but since then we've been spending a lot of time in my lab actually making these types of compounds with different emissive dyes, because there's always a problem to know whether it's the dye that's taking the complex to where the dye wants to go, or whether it's the complex that is going to where it normally goes. And so what we're doing now is varying all of the dyes and varying all of the linkers just to make sure that we know exactly where this is going. But we're excited with these types of compounds because these compounds with these DPPN ligands, we found this particular one has a 17-fold increase in toxicity towards cancer cells, not toxic in the dark, but becomes 17-fold more active when irradiated with visible light. But it's active towards this cell line that is resistant to both doxorubicin and carboplatin. So this could be very useful for treating cancers that are resistant to these tumors. And we know that this works because it makes singlet oxygen with about 60% yield. So that's the mechanism of why this compound works. We also know that it goes to the mitochondria, and this is Amanda. She has done a lot of these cell assays, and I'm not going to go over all of them right now, but what you can see here, this is a stain for the nucleus. You can't see it very well here, and this is a stain for the mitochondria, and you can see that before we shine light, you can see that the mitochondria is very intact, and we start shining light, the mitochondria degrades, and that the DNA actually condenses into this we see this chromatin condensation. And both of these are associated with apoptosis. So this is very nice to see because, as I told you in the beginning, the drug that is currently approved for photodynamic therapy doesn't enter the cells and it's painful to the patient. And so this looks very promising. And actually, Amanda did a lot of other assays that show that the compound actually enters the cells, it localizes in the mitochondria, and induces apoptosis. All right, so the important part here is this DPPN ligand that creates singlet oxygen, and just to put them together now, we also have ruthenium compounds with the DPPN ligand that make singlet oxygen in very high yield. So the question was, can we put these together? Can we have compounds that make singlet oxygen in very high yield and also release drugs, and will they work better? And in some of our first work that we did, we do have a ruthenium compounds with the DPPN for singlet oxygen and nitriles for ligand dissociation. And what we see for this particular compound is an increase in activity of over 1,000 between dark and irradiated, and that is the best, one of the best, if not the best, available today. And for comparison, the compound that just makes singlet oxygen, it only has almost about a 300-fold inhibition. So this is showing us that the compound that can do both things, it can actually kill the cell via 2 different mechanisms, works better than the one that just makes singlet oxygen. Okay. Now, this one again also is delivering nitriles. We also made the compound that delivers pyridine, and so again we have the DPPN ligand for singlet oxygen, but now we introduce methyl groups towards the metal to distort the geometry around the ruthenium to be able to deliver pyridine. And so we have shown now that this compound makes singlet oxygen in almost 70% yield and delivers ligand in about 5% yield. And so this it was a very nice result. Something that we're working on now is to tune these ratios, trying to figure out how to tune them to maybe get them to 50-50. That's something we're working on right now. But we also took this compound and we put a drug on it. So this is imatinib. And so imatinib is currently being used to treat gastrointestinal tumors, and as part of this family of overexpressed enzymes. And we also chose this drug. Again, it has a lot of side effects because it's not localized to the tumor site. But also, these particular tumors make a protein on the outside, and there are antibodies that recognize those proteins selectively. And those antibodies we can buy. And so what we wanted to do is to put this compound on our ruthenium for delivery with light, and also to bind it to one of these antibodies so that the antibody could direct our compound directly to the cancer, selectively to the cancer, so that it would be localized on the cancer when we shine light already, okay? And so we did that, and this is the compound that we made with the imatinib on it. Again, we have the DPPN ligand for singlet oxygen generation and the 2 methyl groups. It makes singlet oxygen with 57% yield, and we do see the ligand dissociation when we shine light. It's very stable. In the dark, you see there are no changes to the absorption spectrum. Then we went on to buy the antibody, and instead of using terpyridine, now we use terpyridine with this carboxylic acid, and we followed normal coordinating techniques, and we bound it to the antibody, and we can measure the ratios of the absorption of the antibody and the ruthenium, and we think we have about, on average, 3 of our ruthenium compounds per antibody. That's a pretty good number for not to bother the activity of the antibody. And once we had this compound, we actually showed that with imatinib, we can actually shine light and deliver the drug. You can see that this is with the antibody conjugate. And so now that we have this antibody model system, now we send this compound to a collaborator in the medical school, because now we need to do animal studies, because now we need to have a mouse with a tumor, and then to show that the compound actually localizes on the tumor, and then shine light and see what happens. So that's where we are with these systems right now. We have a completely new way. Also, now this is antibody selectivity. This is pretty common. We have a completely new way now to also selectively target tumors. And if you're interested in that, you can look at this paper right here that just came out from my group. That's completely different, but I'm not going to have time to tell you about it because I know that many of you have class and other duties at noon. And so I'm going to pretty much stop there. And so I hope that I showed you today that we have a way to shine light to deliver drugs to basically mitigate the toxicity all over the body of current cancer drugs. And now we can combine it with a generation of singlet oxygen to make improved drugs that work together. And so these drugs would not only kill the cancer cells, but would also do something else. For example, inhibit metastasis at the same time. Okay, so with that, let me just thank the students and people that did the work. And so a lot of the work was done by Brian, TJ, and now it's Lauren in my group, and Jessica did a lot of this work, and Malik more recently, and these are collaborators. And again, St. Albert the Great. So, and thank you for your attention.